Welcome back to my A-Level Biology Masterclass, in which I cover biology topics in under 10 minutes. This video we will be going over the topics of genetics, in particular monohybrid and dihybrid inheritances. So please grab a piece of paper. If you listen to my previous videos, you know that I'm a big advocate for writing revision notes using pen and paper. There have been a lot of studies to show that writing notes using pen and paper actually increases test scores. So for my video, please grab a piece of paper so you can make your notes alongside with me. And without further ado, let's get into it. The only thing I ask in return is please like this video, subscribe to my channel, and share it with your friends. Without further ado, let's go over some biology. The topic of inheritance covers quite a lot of different types of inheritances within A-level syllabus, but in essence, it's important to understand that inheritance is the process of how genetic information is passed from parents to offspring. Foundation and principles of inheritance has been originally discovered by Gregor Mendel, and we'll be covering some of his experiments in this video. The different type of inheritances will be included in several future videos of this A-Level Masterclass, so please stay tuned. But I just wanted to summarize them here so you know what the breakdown will be. So in this video, we're going to go through the monohybrid and dihybrid inheritances. Then in future videos, I will be covering codominance. Then I'm going to be covering linkage, specifically for within sex linkage and autosomal linkage. And then the last type of inheritance will involve the discussion of multiple alleles and how they affect the inheritance. And remember, these particular series then will be finished by epistasis and Hardy-Weinberg principle. So let's start by going over monohybrid inheritance. So it's important to understand and define each one of these inheritances so to make this overview clear. So monohybrid inheritance is a type of inheritance where a single characteristic is controlled by one gene with two alleles. And remember, guys, at this point, it's very important to remember that the definition of a gene is that it's a section of a DNA that codes for a particular protein, and the definition for allele is that it's a different version of a gene. But I would highly recommend that you really brush up on your definitions for this topic. Hybrid inheritance probably includes the simplest form of inheritance. An example we're going to use in this video is the color of the pea pod. And before we start, we need to always understand which is a dominant or recessive characteristic in each case of inheritance. So in this case, the dominant characteristic will be the green color of the peat pod, and then the recessive characteristic will be the yellow pod. It's important to understand then when we have a dominant characteristic, as in this case is color green, we normally should go for capital G as notation, and then the little g would be for the yellow pod. This is usually kind of the generic rule, but in exam, you might be told otherwise. So it's important to follow exam instructions in that case instead. Okay, now we're going to move on how to actually work out the genetic problems, and I highly recommend the method of using labels on a side to make sure you don't miss any important information in exam that might gain you marks. So the first label would include saying parental phenotype. So what is the phenotype? How do parents physically look? So in this case, we're going to have a true breeding green pod crossed with a true breeding yellow pod. True breeding is the definition of homozygous. So make sure if you're given this information in exam, that's what it means. The same alleles, two of the same version. Then the next label is parental genotype. So as I said to you, because true breeding means homozygous, in this case, it's going to be capital G, and then cross to little g parent. And the next stage in the label includes writing gametes out. The gamete is where basically one allele is passed in each gamete. And the best way to represent them, in my opinion, is to say, okay, well, you have these two separate letters, and then the gametes are going to be in circles, which shows this very clear to examiner. Okay, I'm running out of space here a little bit, so what I'm going to do is I'm just going to bring this quickly to the next page. Voila, here are we on the next page. So the next label we're going to go through is offspring genotype. This is the label and under which you can then start drawing your Punnett squares to do some working out. So we're going to start with the first F1 generation, so crossing that first cross. And here I'm taking the two parents representing capital G, and little g. So as you can see here, the whole generation of F1 is 100% green pods. They're all heterozygous. So if you're asked to work out the F2 generation, you would take two of those offspring and then cross them to each other again in this case. And I'm going to show you these are the combinations that are possible. And what is interesting here is that we need to look at the phenotypic ratio. Phenotype is how does the organism look? So how are the genes being expressed? So in this case, the phenotypic ratio is 3:1. So we have 3 green pods and 1 yellow pod. And this 3:1 ratio is indicative of monohybrid inheritance. And the final label here is offspring phenotype. So it's a very good practice to actually writing out all the genotypes again. So just make sure whether you're asked F1 or F2 in exam. So in this case, I'm just going to take F2 and we're going to write out all the genotypes again, and we're going to clearly match them to their phenotypes. Quite often I see in exams people just have it kind of all over the place. So you need to make this as clear to examiner as possible. But here's the summary of the monohybrid inheritance for you. Now let's go through dihybrid inheritance. This is the process of inheritance of two different characteristics that are controlled by two different genes. So there'll be two alleles for a particular gene. This is important because it basically shows how genes can actually assort independently. And when I teach my students, I always provide an example of a brown guinea pig with blue eyes. So what is the probability of that brown guinea pig being with the blue eyes? In my opinion, dihybrid inheritance is probably one of the most difficult types of inheritance to actually learn about. There is a lot of confusion, and hopefully I'm going to be able to break this down for you. The extra complexity level will be added with epistasis, so please stay tuned and subscribe to this channel if you want to learn more about that as well. But here I am highlighting some of the key terms. So just make sure that when you learn inheritance, you do actually understand and learn the definitions. So for this particular type of inheritance, we're going to go through an example of a shape and a color of a pea. So not the pea pod as in the previous one, but the shape and color of the pea itself. So again, before we start any genetics problem, it's very important to identify and clarify which are the dominant versus recessive characteristics. So in this case, the dominant characteristics are the yellow color and the round shape, two independent characteristics, and then the two independent recessive characteristics are the green color and the wrinkled shape. And I'm going to use that notation to represent that. And remember, we start with F1 generation. We're going to start using our labels again. So I'm going to write the parental phenotype is going to be true breeding yellow and round, so both dominant, crossed with true breeding plant, where it's going to be true breeding recessive, so green and wrinkled. Then the next stage is we're going to look at the parental genotype. So as we have two independent genes here, which will be represented by the alleles, so we have capital Y, capital Y, capital R, and little y, little r, little r. Gametes is the key skill that you really need to understand in this topic. So in this case, the one allele will be representing one each characteristic. And remember, they are inherited independently. So one of each letters needs to come and be present in the gametes. So in this case, it's going to be capital Y, capital R, and in the second case, it's going to be little y, little r. The only possible combinations here. That's why I'm not drawing any other circles. Okay? So when we combine these together, the only possible offspring genotype in this case is going to be capital Y, little y, and then capital R, little r, which will be representing the F1 generation offspring genotype. And in this case, the offspring phenotype is going to be yellow, and round 100%. So that finishes the F1 generation. But what we're going to do now for the F2 generation, we're going to take the offspring genotype from the F1 generation and cross it together. So to simplify the matter, I'm not going to use the labels anymore, but for you guys, I ask you please copy over the same label and draw it out properly. So let's now start going through the F2 generation. In this case, the parental genotypes are going to be as follows. So if you copy this over, and the key skill now comes in deciding what are the gametes. So the rule is one of each letters in each gamete. So here I am drawing all the possible combinations when the letters are different. So let me write this out. So it's capital Y, capital R, capital Y, little r, then little y, capital R, and then little y, little r. Okay, so really understand this step. It is the key and where the most mistakes happen in exams. So really practice this out, because when I teach this, I see all sorts of combinations. So get this right. Okay, this is where the difficult part now will come, because we now need to cross all of these gametes and see what are the potential combinations. So for this, you need to draw this big Punnett square, which is also similar for the epistasis. So I'm gonna write all the gamete options for each parent on both of the sides. Now please pause the video and write those combinations out by yourself. For all the combinations, I hope you got them correct. And now let's go through it, because again, we need to work out that phenotypic ratio. So I'm going to start with recessive characteristics. I'm going to do the wrinkle shaped here, represented by a little squiggle. So these are the 4 genotypes that will have a wrinkle shape. So it's 4 of those. The next step is then to identify the recessive color of green. So where the little y are, there will be 4 options here, as I'm highlighting them in green color. And this should give us enough information to now understand and work out the phenotypic ratio. So let's go for it. So let's count it. So to get the yellow and round shape, so dominant characteristics, there are 9 in total. And I'm going to show them here by a laser pen. So these ones. Then the next phenotype is going to be round and green. So again, I'm going to show them here by the laser pen, and you can match them to your own notes. So that gives us 3. Then the next one is going to be wrinkled and yellow. And again, there are going to be 3 as shown here. And then the final one is going to be wrinkled and green. So both of the recessive characteristics inherited independently, and it's this one over there. And that basically is how you work out the dihybrid inheritance in exam, and the phenotypic ratio is 9:3 to 3:1. I hope you really enjoyed this video. Please subscribe to my channel, and I will be releasing more videos like this. And please comment below what the next videos you'd like to see. I really appreciate your support. Good luck with your revision and exams, and I will see you in my next video. Goodbye.